Okay, we are going to, here we go, talk about gene therapy. So I like this cartoon. I think it's kind of funny, but that's not exactly what gene therapy is. So let's talk about what is gene therapy. So the idea behind gene therapy is that you're trying to replace a mutated gene that causes disease. And you're trying to replace that with what we call a healthy copy or a wild type. Replace mutated gene that causes disease with a healthy copy. So say you have a mutation in a protein that's not producing a functional protein, then, excuse me, you would try to get in the right gene sequence and so that the cells would make that correct protein. Another option is you can inactivate or knock out a mutated gene. So maybe you have a gene mutation that's causing a protein to cause the disease, but you don't really need that protein to be made. So maybe you can just inactivate kind of the bad player. Most of the time, though, we need to deliver some type of healthy copy to help fight the disease. So I guess the difference between 1 and 3 is you might be able to use CRISPR Cas9 here to, maybe I shouldn't have used replace, but to repair a mutated gene. Or you might just deliver a brand new gene sequence that overrides that defective protein. So it's based at the molecular level, so it's different than a drug. You're really actually trying at the molecular level to make a more permanent fix to whatever's causing the disease in the person. So there's a lot of challenges to this. The first thing is gene therapy is best for what we call monogenic diseases. So diseases caused by one gene. Right. So if you have 5, 6, 7 genes that are all, say, in a pathway or in multiple pathways that are causing disease, it's going to be really hard to try to fix and change 5 or 6 genes. But if we only have one gene to deal with, we have a much better chance at success. It's also best for if the disease is in a specific cell or tissue type. So you'll see some examples for cystic fibrosis, which affects the lungs, or blindness, which affects the eyes. So something that is localized is much easier to target. And then a big goal is it to be long-lasting, so you don't have to have repeat treatments, but that requires either manipulating the genome, like with CRISPR, or integrating a healthy copy into the person's genome, right? So whatever way you're doing this, you are making changes usually at the DNA level if it's going to be long lasting. And we'll talk about some types of gene therapy that aren't long lasting, but may be the best alternative. A lot of your challenges is really in the efficacy of delivery. So can you get, sorry, I can't write and talk this morning. Can you get the gene or the CRISPR or whatever you need to manipulate to the proper cells? You want to be sure the gene of interest, if you're putting in a healthy copy, is expressed and is expressed at the right levels. So there's no good in putting in a new copy of a gene if it doesn't get expressed and make enough protein to help with the disease. Whoops. I don't know why it does this. Potential issue with the integration into the genome is that you don't want to disrupt normal genes. And we'll talk about this issue. And then we're going to talk about that a lot of these are based on viral vectors. And so you don't want to trigger an immune response. You don't want your body reacting or the patient's body reacting and rejecting the treatment. So we're going to talk about some general characteristics of gene therapy, and then we'll go into some specific examples. So really you have two options for delivery, direct delivery or in vivo. So you're directly delivering it to the patient, or cell-based, which is we call ex vivo. And this is where you remove cells, modify, and return them to the patient. So in vivo, like it says, direct delivery of the gene therapy or ex vivo out in the lab. So let's look at these two options. So in vivo gene therapy is really hard. So I don't know if I have my image. Okay, it's hard to deliver. So the challenge is delivery and targeting and avoiding that immune response. And we're going to discuss these more in a minute. But what this image is showing you is some of the most common ways for in vivo is DNA in a liposome. So this is kind of like the Moderna, Pfizer vaccines where they're delivering RNA with a liposome. Delivering things directly into the tissue. And you can do all these in different combinations, right? So you could use a tissue injection with liposome or infusion into the bloodstream with a liposome or with a recombinant virus. So the virus would be shuttling the genetic information. There are some potentials with a biolistic gene gun. This is kind of like the electroporation idea of jamming that DNA, plasmid DNA into the cells. But as you can imagine, this is only going to work on surface cells that we can get to. Ex vivo gene therapy is actually what's being done the most right now. And so what you're doing is you're removing patient cells you're manipulating them in the lab. And that can be done, again, through things like DNA liposomes, through electroporation. And then you're usually growing them back up and putting them back into the patient. So the plus of the advantage of ex vivo, the positives, is it's not likely to trigger an immune response. And you can check the cells. I'm just writing all over here. You can make sure that the cells are healthy. You can make sure and they're expressing whatever gene you're putting in there before you deliver it back to the patient, right? So it's not as blind. But the negatives are that this is mostly targeting stem cells, and so that can't fix all types of diseases. So you're limited. You're limited by, I guess, that one, disease type. Right. So if stem cells aren't going to work to help this disease, then ex vivo gene therapy might not work as well. Or if they're not cells that you can take out, right? So if you're trying to fix someone's liver, can't really pull out those cells, fix them, and put the liver back. So there's challenges with ex vivo. So the next slide I just wanted to show you was what are the common vectors. So remember, vector is a way to move things around. So you can see most of these are virus-based vectors. Oops, plasmid DNA is coming up. Lipofection we'll talk about, and then some more virus vectors. Let's talk about naked DNA and lipofection first, since they're not as common, and then we'll get into comparing the different viral vectors. So, as the slide showed before, you can use naked DNA, which is your plasmid, which you know about from gene cloning, or you can use the DNA wrapped in a liposome, or the liposome attached to, or sorry, the plasmid attached to the liposome. So liposomes are just artificial membranes, sorry. And most of these are given either via an intramuscular injection or through an intravenous route. So the positives of these types of gene therapy is that they're not pathogenic. So there's no virus involved. You're not going to cause any disease. There's no size limit. We're going to talk about size limitations and size of genes in just a couple slides. There's no immunity issues, at least not that they've come up with yet. And it's pretty, I'm going to put asterisks. It's inexpensive in the sense that it's really cheap to make up lots of DNA and to make up liposomes. But the negatives, and when I say inexpensive, this is for the materials, not for the putting it into the patients themselves. The negative is, like I said, this ballistic gene gun is limited to the dermal tissue. Oops. They both have very low efficacy, although I'm wondering now with some of the new ways they're manipulating the liposomes for the mRNA vaccines, if this will be worked on. It's very hard to target specific cells with either of these techniques. And there's a very low rate of integration of the DNA. So it's not usually permanent. Right? So when I say integration, what I'm talking about is, would be ideal is if here's your chromosome and we can insert that new gene sequence, we can integrate it into your chromosome so it becomes a permanent part. So lots of things to weigh out, pros and cons, when you start talking about gene therapy vectors. Let me show you an example of a naked DNA application, gene therapy. So you may recognize this is Blake Lively and her dad. And her dad in 2003 had a heart attack, which eventually damaged about half of his heart. So he was working with half heart function and just didn't have hardly any energy. So in 2013, he went to the Utah School of Medicine, and they used a catheter to access the cardiac vein, and they infused, so they delivered naked DNA, so plasmid, that expresses the SDF1 gene. And SDF1 codes for a protein that's a chemoattractant. So it's a chemoattractant for stem cells. So the idea is you give these cells in the heart this SDF1 gene overexpressing. They express this chemoattractant. Your own stem cells come and repair the heart. And I've given you links to, if you want to read more about these. I know I'm just going to kind of throw a lot of different examples at you. But 7-8 years later, he's still alive, doing fine. And I'm trying to remember, I can't remember how many people they have done this on, but it's been a fairly successful, let's see. It said they were working, sorry, as of, yeah, my data is a little old. As of 2013, they had at least 72 patients that they were trying this on. So this is a nice successful example of gene therapy. I couldn't find follow-up information about if they even go back and test to see if that gene is still there, or if the fact that his heart has healed up, that's the measure of their success. So I mentioned that with naked DNA and lipofection, you don't have a size limitation. But when we start talking about these viral vectors, we do have a size limitation. And so how big is a human gene? Well, the genes themselves are big, 10 to 15,000 base pairs. But then we have to remember that most genes have the introns spliced out. So the messenger RNA is usually what we're talking about. And remember, we talked about this with recombinant DNA using the messenger RNA. And those are much smaller, around 3.5 kilobases. But there are some that are big. So for instance, we're going to talk about the cystic fibrosis gene, and it's a pretty big one. Exon length means the mRNA, 4.4 kb. So the size of the gene is going to affect what size or what virus vector you can potentially use. And dystrophin is one that they've been trying to use for muscular dystrophy. And that's huge. That's 10,500 base pairs. Look how long that intron is. Isn't that crazy? We splice out so much of our DNA. I think it's really interesting. But that's a molecular biology course. So we've talked about viruses. Just to remind you, I mean, a virus is basically genetic material in a protein coat. And yes, some viruses get more specific than that or more complicated. But the viruses we're going to talk about, you've got a protein, which we call capsid, and you've got your DNA or RNA in there. Now, some viruses have an envelope around the outside, and some of those viruses are used. So like we've talked about, coronaviruses have these spikes and envelope, and then what's inside is the DNA or RNA with its little protein coat. Okay. So why would we use a virus? Well, viruses are really good at targeting cells. They have specific receptors. And so you can use a virus that's going to only infect, say, liver cells or muscle cells or heart cells because of the receptors and the way the virus interacts with a receptor. That's my little receptor here. Viruses are basically genetic delivery machinery. So they're really good at delivering genetic information. That's what they do. That's how they replicate. Some can integrate into the genome. So retroviruses like HIV, the reason once someone is infected with HIV, they have HIV for life, is that it integrates into their genome and becomes part of their chromosome. This is great if you want a permanent fix. And viruses can be modified. So we can remove the parts of them that cause disease. We can use just the shell, or the shell and the capsid in the envelope. And we can manipulate even these spike proteins to make different viruses target different types of cells. So viruses are great at delivery. The big negatives is the potential for an immune response, because when our body sees something foreign, it's going to try to fight it off. And like I keep mentioning, size, or what we call packaging limitations. So how much genetic information can that virus hold and deliver? So this next slide just shows you, again, those viruses used in gene therapy. So we're going to talk about adenoviruses, retroviruses, which include the lentiviruses, and we're going to talk about AAV, adeno-associated virus. You can see that herpes and pox viruses are used at a little lower efficiency level, vaccinia virus, but the main ones you're going to see are the adeno and the retroviruses, and the AAV, and we're going to talk a lot about AAV. So this is to give you some characteristics of these viruses. So adenoviruses are nice. They have a big genome. They can carry about 7.8 kilobases of transgene or whatever new genetic information, but they don't integrate. So they're not a permanent fix. Adeno-associated viruses have the positive that they do integrate, but they have about a 4.5 kb size limitation. So they can't carry very big genes. Retroviruses have a good 8 kb size limitation, but they can only infect actively dividing cells. So that could limit your target population. Lentiviruses are a subtype of retroviruses. You can see that they have a similar size limitation, but they can infect dividing and non-dividing cells. And I will add in here the herpes virus are really good at targeting neurons. That's what they target. And neurons are very slow or not dividing at all. So if you're going to target a neuronal disease, a lot of times they'll use herpes virus. The benefit of retro and lentil is that they both integrate into the genome. So they can be long lasting. And then we talked about liposome and plasmid. The main is the unlimited size. The main advantage is the unlimited size. So you can put in lots of DNA information. They can go to technically any cell. The problem is the efficacy of it. So this is just a slide that kind of summarizes this information. Some of it's a little bit different than that other slide, but just another reference for you. I want to talk about adenoviruses because they're one of the most popular ones being used. And mainly adenoviruses are so popular because it's easy to manipulate the outside. And that means you can target different tissue. So they're great for changing the targeting different tissues. The problem is they don't integrate, so they're only a temporary fix. Retroviruses, again, lentiviruses can integrate. This is just showing you. So you have a permanent fix, but we're going to talk about some of the issues with that. Ideal gene therapy. I'm looking at my notes. We already talked about this back here. You want to target specific cells. You want it long lasting. You might need to look at, back here we talked about monogenic disease, and you don't want to trigger an immune response, and you don't want to disrupt other normal genes. So where are we? Here's some of the things that they are looking at. A lot of gene therapy is targeted to cancer. We're going to be talking more about gene therapy to target diseases, monogenic diseases, and inherited diseases. But you can see all these different types up here. Just so you know, this is kind of a cool website. You can access it here, and it gives you all of this data, and then you can click, there's more links below to see like what kind of cancer clinical trials are going on, or what kind of cardiovascular monogenic clinical trials. You can also click on this, and this will take you right to the US clinical trials, and you can look at who's recruiting and who's not, and what are they looking at, and some even have information on how the gene therapy is being delivered. So pretty cool resources. The other thing this slide just shows you is what are some of the genes that they're trying to do. So a lot of it is receptors or antigens. This is really important for the cancer gene therapy. So you want to put something in the cells that attracts the immune system to target them and kill them. When I first saw suicide, I was like, what? But this means a suicide gene. So you put a gene into a cell and it basically makes the cell self-destruct. So just a little bit more information on what research is looking at. And this is worldwide gene therapy trials. Oh, so it's right there. Okay, so let's talk about some of the history of gene therapy and some of the highlights and what I call lowlights. So this young woman, Ashanti De Silva, in 1990, you can see her here. She was 4 years old, and she's holding hands here with W. French Anderson, a very famous gene therapy molecular biologist. And her and this woman, Cindy Cystic, they had a disease called ADA SCID. And SCID stands for severe combined immunodeficiency. The ADA stands for adenosine deaminase. And SCID is severe combined immunodeficiency, which means they had an immunodeficiency disease. And this is normally a fatal disease in childhood. It's autosomal recessive. And if you don't get treatment, basically you don't have an immune system to protect yourself from disease. So what Ashanti got, it was an ex vivo gene therapy. They transferred the ADA gene into her T cells in the lab, and then they put them back, re-injected them into her. And this treatment was repeated for a couple of years. She was also getting some protein, ADA protein therapy, replacement therapy. So she wasn't just getting gene therapy, but her modified T cells persisted for about 10 years. Cindy got this same treatment at age 10. And you can see that they both grew up into healthy, happy adults. They're now screening for this disease even earlier and using umbilical cord cells for newborns and doing the gene therapy. And it's down to like a one-time treatment to help with this disease. So this was really cool. What did I put? Oh, yeah. So it was a retrovirus. So like I said, retroviruses are great because they can integrate. And so if you can permanently fix that population of T cells that helps with the immune response, you have a successful gene therapy. Big issue. Oh, I'm going to jump to this one and then we'll come back to Jesse because I want to go on with that idea of retroviruses. So gene therapy, in 1976, John Travolta starred in a movie called Boy in the Plastic Bubble, based on this young guy, David Vetters. So he also had a type of SCID, a severe combined immunodeficiency, and the only therapy at the time was a bone marrow transplant. And so they tried to do a bone marrow transplant with his sister, who wasn't an exact match, but he ended up dying about 4 months after the bone marrow transplant due to a lymphoma, a cancer, due to Epstein-Barr virus, which is the virus that causes mono. He somehow got that infection during the transplant. So this wasn't gene therapy, but he made this disease very popular. And sorry, this is X-linked SCID. So I put him up here because he kind of, NASA even built, in this school, built this bubble for him to live in so that he had basically clean air and wouldn't catch any diseases. So this has been a target for gene therapy. And in 2003, they did the first gene therapy trials. It was ex vivo, and it was bone marrow. And they used a retrovirus to do the gene therapy. They tried this on 20 children. And 2.5 years later, 4 of the 20 children developed a T-cell leukemia. And what they found was that this retrovirus had inserted the gene into a cell that, into a gene that ended up causing the cancer. So this was a huge setback for gene therapy because retroviruses were looking at as a very good potential vector, but we can't control where they integrate the DNA into the cells. And it seems that they target parts of the genome that can be cancer causing. The other 16 children are doing fine, but 20% of them got disease. So another kind of low light for gene therapy was Jesse Gelsinger here. This happened in 1999. Jesse had a somatic cell mutation, so his wasn't an inherited disease, but a spontaneous one in this gene called OTC. It's an X-linked genetic disorder of the liver for ornithine transcarbamylase. And so he actually produced some normal protein and some mutant protein. So he was able to be a healthy person if he had special diet and some medications. If you have the total disruption of this gene, it's fatal at birth. So he wanted to give back to the community. So as soon as he turned 18, he signed up to be a healthy patient in an adenovirus vector clinical trial. Okay, so they're using adenovirus to deliver the OTC gene. His immune system overreacted. He actually ended up going into a cytokine storm, and he went into organ failure, and he died within 4 days. So he had one of these rare but horrific reactions to the gene therapy, and this became a huge setback for the field. So all gene therapy trials were stopped, and went back and kind of re-looked at this. But we kept going on. This disease, people may have, this is another X-linked disease, There was a movie in 1992 called Lorenzo's Oil. In this disease, ALD, the people cannot degrade fatty acids, and it damages their myelin sheath, which is that protective coat on your nerves. And it results in mental disability and usually death at the age of 6 to 8 years old. So the only effective therapy is stem cell transplants, which again, anytime you say the word transplant, you require a donor match, right? So we can't use stem cells from this own person because they have a gene defect. You have to diagnose this very early. And so in 2017, there was another ex vivo, see a theme here, gene therapy trial where they took the bone marrow from the patient and they used a lentivirus vector and they put in this, it's actually called an ABCD1 gene, and changed or altered the patient's stem cells, had to do chemotherapy to get rid of all of the remaining bone marrow cells, and they put the cells back into the patient with an IV, and 15 out of the 17 patients have stable neurologic functioning 2 years past this gene therapy. And I haven't seen anything further, but this was a pretty good success. There are other gene therapies you can find looking at this disease, but this was one of a nice success. Again, ex vivo, right? Using stem cells, manipulating them in the lab. This one I brought up because it's being worked on currently. This is Parkinson's disease. Parkinson's disease is looking at, you can see the gene therapy for Parkinson's is looking at AAV, adeno-associated virus, which remember I said is a small virus, but it does integrate. It can cross the blood-brain barrier. And so what they've been looking at, and there's some review papers here, is putting these different genes into the AAV vector and injecting them directly into the brain. Because what's happening in Parkinson's is you're getting degeneration of neurons that produce dopamine. And so that loss of dopamine, which is a neurotransmitter, results in issues with regulation of body movement. So people with Parkinson's have tremors and repeated movements and walking difficulty. They talk about masked facial expressions. This neurotransmitter is involved with memory, reward, motivation. So there's mental and behavioral issues, pain, GI. They say 6 million people worldwide are affected by Parkinson's, 1 million in the US. There are medications, but they become less potent as the disease progresses. So this is an area which is having a lot of work in Parkinson's, partly because we've got that tissue that we can specifically target. Again, a monogenic disease, and we can target a specific area of the brain with these injections. So you can look more into that. It's looks like some promising results. All these sound great, and we'll talk about one of the reasons, which you probably can guess, is money, that we're not curing everything right now. Another one I just wanted to point out is that's really has a lot of gene therapy research going on, and here's some brand new resources, is cystic fibrosis. So cystic fibrosis is a disease in the lungs where the person is not producing this chloride transporter. And so their lungs fill up with mucus. They can get a lot of infections. And so cystic fibrosis is one of those nice targets because, again, it's a monogenic disease. It's targeting one specific organ, the lungs. It does affect sweat glands in the gut also, but if we can get the lungs fixed, the person has a much better chance of living a more normal life. And so this is just showing you that there's lots of work being done for cystic fibrosis, adenoviruses, AAV, hoping to integrate. The problem is that the cystic fibrosis gene is right at the limitation size of what the AAV virus can package. And so they're trying to see if they can make the gene a little bit smaller, because you still need to get the promoter in order to drive gene expression. They're looking at, this is Lentivirus methods. Looking at DNA, liposomes. This has not been very efficient, and they're not getting enough material in there to really have enough, I guess I should say, enough normal protein or healthy protein to help fix the disease. But cystic fibrosis is one of those gene therapy targets that's been worked on for a long time, and they're making some really good progress with it. This I bring to you because it's the first gene therapy that was actually FDA approved. This is a genetic inherited form of progressive blindness. It affects about 1,000 to 2,000 people in the US. They are using, again, an AAV vector to add the gene back. It doesn't seem to be a cure. So far, they've had improvement for about 4 years. We don't know if it's going to work forever. But there's a mutation in this gene that impairs the eye's ability to respond to light. And so you end up with destructions of the photoreceptors. What I found interesting, just kind of an FYI, which leads me to some of the next slides, the cost is $425,000 per eye, $500,000. So you can imagine that in clinical trials, you usually get this for free, but if this becomes a therapy or option, not a lot of people are going to be able to afford it. This talks about the risks and how it works. So if you want to learn more about this type of gene therapy, check out those links. A gene therapy paper that we read in Biotech one year was for this treatment of spinal muscular atrophy. So this is a disease that basically causes children to be in wheelchairs, their muscles atrophy, and most children with this don't live past the age of 2, or they have to be on breathing and feeding tubes. Cost. Oops. $2 million for one dose. And this is, again, another AAV vector, so it may integrate, but can you imagine $2 million? This affects about 1 in 10,000 children. And the company that had developed this said that, at least in 2019, said that they were going to give away 100 doses via a lottery. They had production limitations on making this. And so if your child had this, you could sign up for the lottery and hopefully get one of the 100 doses. Crazy. Another one that's also important, this I had read, this is for sickle cell and beta thalassemia. Make sure I spell this right. Thalassemia. Basically two types of, I hope that's right. Two types of anemia. They said the cost for this treatment was $1.8 million. Although it may have come down, this was from 2019, I got that number, and found a more recent article. This is also an ex vivo approach. One is using CRISPR. They've tried this on 19 people so far. They're using CRISPR to target and shut off a gene. And the reason they're shutting off this gene is this gene shuts off fetal hemoglobin production. So apparently as you're developing, you produce fetal hemoglobin, and then as you get to a certain age, that gene is shut down and you switch to adult forms of hemoglobin. Hemoglobin is the molecule that carries oxygen to all your cells. So sickle cell disease has mutations in the hemoglobin that makes the hemoglobin clump up, and so it can't deliver oxygen properly. And the cells sickle, which means they turn into kind of like a sickle moon shape, and they can get stuck in small capillaries. And apparently it's a very painful disease if you have, if you're homozygous for the sickle cell. Both of these require blood transplants. The only cure is, again, a bone marrow transplant, which means you have to find a donor and all of that. So what they're doing with this ex vivo is they are taking out the stem cells, hematopoietic, the bone marrow from the patient, fixing them with CRISPR, and then putting them back into the patient. So this is the first published account of gene editing, and you can go to the Nature paper. This is just kind of the background paper, and then it will link you to the real paper. This is the first published account of gene editing to treat a heritable disease. And they're trying some other types of gene therapy for sickle cell and beta thalassemia using other vectors. Oh, what I wanted to say is, so just so you know, they're doing electroporation to get the CRISPR system into those hematopoietic stem cells, and then growing them up and then putting them back into the patient. So in all the years I've been teaching biotech and biology, gene therapy is one of those ones that's really cool and promising, and it's just taking us a long time to figure out how to fix some of these genetic based diseases. So this is kind of where we're at right now. You can see that most are in phase 1 clinical trials. Very few, 0.1%, have made it to phase 4, a little more in phase 3. And just to remind you what that all means, the phase 1 is where you use healthy individuals. So we're looking at safety of the medication and the treatment. This is the one where Jesse Gelslinger died, and so that gene therapy trial was stopped very early on. Phase 2 is you're looking at efficacy. So now you start bringing in patients with the disease and trying out your gene therapy. Phase 3, you're tweaking it even more. So safety, efficacy, and dosing. So you're trying with different participants, and these numbers are ideal, and this is really for drug therapy, because you might not even have 1,000 patients with this disease for a phase 3 trial. And then phase 4 is looking at long-term effectiveness, cost effectiveness, success. So gene therapy, I think, is a super exciting field. Lots going on, lots of challenges, and we will be reading a paper together about a current gene therapy, not trial, but technique that's being researched right now. All right, thank you. Let's see if I can get out of here. Back to my Zoom. And we will stop.